I'm Harpreet.
I'm Lucy.
I'm Edith,
and this is my Dice Talk.
As you may know or may not know, we hold Dice Talks once a month. And so today is our October Dice Talk. And in a few moments, I'm going to introduce our three speakers. So we've used this month's Dice Talk to showcase some of the research that has been done by our taught MSc students who have just completed our taught MSc in Conservation Science, but also some students on our Masters by Research program as well. So I'm going to introduce those speakers and thank them. We're going to save questions at the end. So we're going to have a series of 3 short talks, and then we'll have questions. I'll ask the 3 speakers to come up and field questions at the end. Okay. So thank you all for coming. Just a few points before we start. So as usual, we have a Dice. We have a presence on all the social media platforms, and in particular, we have a QR code there for the Dice newsletter, and that is where we make all of our announcements that Dice has in terms of its engagement and civil mission in sharing the message about the conservation work and research that we do all over the world, but also close to home as well within the UK and the activities we have and so on. So do take a moment to check out our newsletter, which is online. Okay. So, and before I introduce the first of our 3 speakers, also just to make a note of next month's Dice Talk. Oh, 2 weeks' time. Okay. Wow. 2 weeks' time is on the 16th of October, which is a talk by Dr. Michelle Agnostou. And Michelle is going to talk to us about understanding complex criminal networks for nature conservation. So do make a note of that in your diaries for the 16th of October, which happens to be my birthday as well. There you go. Anyway. Okay.
Good evening, everyone. My name is Harpreet, and today I'll be sharing about my dissertation, Assessing Wildlife Stowaways, Pathways and Risks in the UK under the supervision of Dr. David Roberts. So the topic itself begs us the question, what are wildlife stowaways? So in easy language, wildlife stowaway refers to any organism that transports from its native region to a foreign environment attached within or to a transport vector. And it can lead to harmful environmental impacts, public health issues, and many other economic cost damages as well, where it is introduced. All these species you see here, be it the Indian saw-scaled viper, be it the Balkan lizard from Kos, be it the clouded monitor from Thailand, that's what they share in common. All of them have entered the UK as stowaway at some point. So globalization is expanding trade and travel networks, which leads to enhanced global connectivity, which also unintentionally facilitates the increase in wildlife stowaways, which is an emerging pathway. As a result, not only the number of alien species are increasing, but the ecological, public safety, and economic costs associated with their impacts are also rising. In a study by Turbulin, he found out that the species introduced as stowaways generated an economic cost of $144.9 billion across the globe. In the UK particularly, the high human population, major ports, airports, particularly in the south, result in the most introduction occurring in this region and subsequent spread largely northwards. Holidaymakers, tourists, travelers appear to be the largest contributor of stowaways in the UK. The wholesale and retail supply chain seems to be a bit underreported. Alien species are also a source of disease transmission to the native species. Emerging pathogens include BD and BSal, which are capable of surviving British temperatures and could pose a serious threat to native amphibian populations. We'll be talking more about it as we go forward. So overall, the primary literature for wildlife stowaways, be it across the globe, be it in Britain, UK, all of them seems to be very scarce. Limited data on frequency, pathways, and effectiveness of biosecurity measures leaves a gap for comprehensive research, an attempt which I made through my dissertation to complete. My research basically focused on these 3 questions. The first one being, what are the most commonly intercepted species across the UK, and what are the key characteristics of these introductions? What are the dominant pathways for wildlife stowaways in the UK, and which entry points are at the highest risk? The third question focused on what are the risks associated with wildlife stowaways, and how do trade and travel networks influence these risks? As parts of my method, I used the mixed method approach. I started with collecting data. So I used purposive sampling to reach out to 11 organizations in total. Two of them replied, the National Center for Reptile Welfare and RSPCA. Then I also reached out to every council in the UK using Freedom of Information Act, and I got responses from 14 councils, 13 of them met the inclusion criteria. I also use a keyword-based search to have a look at the news reports about wildlife stowaways across UK. Overall, I was able to compile a data set of 1,025 records of wildlife stowaway interceptions across UK between a decade starting from 1st January 2015 to 30th Jan 2025. Now, for data analysis, for my first question, I used descriptive stats to look at the most common species, as I mentioned. Then I used multinomial logistic regression to formally test which characteristics were most influential when it came to wildlife stowaways, with my response variable being class. And when I say class, I refer to reptile, amphibian, inverts, which was what my study focused on. For the second one, to look at pathway and entry point risk, I used a combination of descriptive stats, multinomial logistic regression, and network analysis. And finally, the qualitative part of my study, the literature review, involved assessing and looking at the risk associated with wildlife stowaways in the UK. So, what did I find out? So, out of the 421 emails I sent, only 14 replied back with relevant data, 13 meeting inclusion criteria of around 68 interceptions across that decade, which seemed pretty low. But as you can see, most of the councils didn't have the data, which points out towards the lack of data, as I mentioned earlier. Then, as you can see, there's an upward trajectory from 2015 to 2024 with 7 interceptions in 2015 and 264 in 2024 with a slight decline. And as you can see, the confidence intervals seem to widen up, showing uncertainty. This can be due to because 2025 data is not really complete, and also a lot of the cases are actually underreported. In terms of class, reptiles were the most frequently intercepted class. For types, lizards, spider, frog made the list. I was able to narrow down 496 species down to their species level, out of which included 164 unique species, and these were the most common ones, the Moorish gecko being the mostly intercepted species. So, when it comes to mode of transport, air was the most prominent mode of transport, with personal luggage being the most prominent means of arrival. Spain, Italy, and India were the main sources of origin from where these stowaways are coming, and that's particularly in England. We'll discuss why it's England. And it's coming particularly to the counties of Greater London and West Yorkshire, with considerable variation among other counties. So, looking at what my models basically show. So, this particular model was created using multinomial logistic regression, and I use a stepwise modeling approach to look at the most influential characteristics. These three were mode of transport, means of arrival, and commercial association to that trade. Yeah, sorry for that. Of these, commercial association was the only statistically significant factor for amphibians, showing they were significantly associated with commercial rather than non-commercial pathways. Mode of transport and means of arrival were not statistically significant predictors for amphibians or inverts likelihood relative to the reference category. In this case, the reference category is reptiles. So if we look at this plot, which shows predicted probability of stowaway across three modes of transport visited by commercial association and the means of arrival, reptiles consistently showed the highest predicted probability of interception across all modes of transport and means of arrival, with amphibians generally the lowest and inverts somewhere falling in between, with quite prominent interceptions for inverts for the other category, be it for commercial association or not. To explore these relationships further, with particularly emphasis to entry pathways, I conducted univariate model logistic regression for mode of transport and means of arrival. For particularly means of arrival, amphibians were significantly more likely to be intercepted through fresh produce compared to personal luggage, while inverts also showed strong association with fresh produce and the other category. Shipping containers, however, did not show a significant effect for either amphibian or inverts, with amphibian estimates in particularly being quite unstable due to their low numbers. For mode of transport, neither road nor sea transport significantly changed the odds of interception from origin. For mode of transport, neither road nor sea transport significantly changed the odds of amphibian or invert interception compared to reptiles relative to air. But the baseline terms confirmed that reptiles were consistently and significantly more likely to be intercepted than either amphibian or invert via the air transport. From there, I moved on to network analysis using a Sankey diagram. I visualized the flow of interception from origin countries to UK destination, stratified by mode of transport and means of arrival. So particularly for this one, air was the most dominant pathway into the UK, with highest interceptions from Spain, Greece, and Mexico. For sea, it was US, China, and Italy, and for road, which wasn't very often, but it was Italy, Spain, and France. So when looking at means of arrival next, personal luggage was dominant, particularly from Spain, Greece, and Mexico again. Fresh produce was most linked with interception from Italy and Spain, while shipping containers were strongly associated with India and China. Other pathways, though smaller, showed notable contributions from countries such as China, Spain, and the US. So overall, the results show a clear pattern in both biological and geographical dimensions of stowaway introductions, with the reptiles dominating interceptions, air and personal luggage representing the most common pathways, and specific countries acting as repeated sources. So, what does the wider literature say about it? The wider literature says that unintentional transport of alien species is a major global biodiversity issue, and the real impacts on and creates real impacts on biodiversity, public health, and trade. Interceptions have increased sharply since 2015, suggesting a growing biosecurity challenge. Amphibians were particularly linked to commercial routes like fresh produce, aligning with the wider literature, pointing out to live vertebrates, hitchhikers, and agriculture and ornamental plant trade. Additionally, in comparison to amphibian, reptiles are more tolerant of temperature fluctuation and diseases, so which led to lower mortality and also higher number of interceptions. Inverts, on the other hand, were even more common, particularly through fresh produce and well adapted to survive trade pathways. When we look at pathways, air transport was the most important and most significant pathway for interceptions. Over the years, passenger planes have increasingly brought in alien species, and with air travel expanding in recent years, the risk seems to grow. Spain and Italy stood out as major source countries, though they also linked tourism along with being major exporters for fruits and vegetables. Within the UK, most interceptions were received in England, especially in Greater London. The reason is that London is a major gateway with Heathrow and major ports handling most of the country's traffic. London is also a hotspot for stowaway because urban areas can provide warmer microclimates. Looking beyond air travel, sea transport remains a major concern as well. Ports act as invasion hubs through ballast waters, hull fouling, or even species hidden in cargo. Zebra and conger mussels are good examples for that. Once introduced, they form dense colonies, disrupt ecosystems, and cause serious economic damage. India was the biggest contributor by sea, mainly because of the stone imports, which are well-known pathway for lizards and inverts. Italy, on the other hand, was important for road transport and fresh produce. Historically, this risk is not new. The yellow-tailed scorpion likely arrived in the 1800s in Italian masonry and now has a thriving population in Kent. So overall, while air dominates, sea and road also play an important role, with each pathway linked to particular countries and species. This shows the need for pathway-specific checks and interventions rather than treating all interceptions the same way. Lastly, then comes the risk associated with wildlife stowaways. For public safety concern, most interceptions are first detected in personal luggage by the members of the public. This creates a clear risk because many people are not really equipped to recognize or safely handle potential hazardous animals, and there is also threat of zoonotic diseases. For ecological risk, one of the greatest concerns is disease transmission. Amphibian fungal diseases such as chytrid caused by BD have already driven global declines. And also bioclimatic modeling consistently identifies UK as highly suitable for BD survival and spread. Out of the 7 native amphibians, 6 breed in permanent water bodies, which is also suitable habitat for BD. A related emerging pathogen is BSAL, which has caused mass die-offs of newts in Europe and could pose a severe threat to species like the great crested newt in the UK, which is highly susceptible to it. Also, interestingly, the peak activity periods for the great crested newt, which is March to August in England and Wales, coincide with temperatures compatible for B-cell growth, suggesting that climate is unlikely to limit the pathogen spread. Along with disease transmission, competition with native species is another pressing issue. Non-native reptiles and amphibians may compete with local populations for space, basking sites, breeding territories. There is an example of an interaction between an invasive common lizard and a sand lizard, and how common lizard was able to attack and maintain its hold over the basking spot against the sand lizard in Dorset. These pressures, combined with climate change and the possibility of further introduction, raise the risk of localized or even national declines in vulnerable species. So overall, Beyond the obvious transport pathways, the risks are very real for both people and wildlife, from zoonotic diseases to invasive competitors and emerging pathogens that could reshape UK's ecosystem. So what can we do? At the moment, control measures for alien species are mostly reactive. In other words, they are only implemented once a species has become a problem. By that stage, management cost is very high, and that requires extensive ecological, economic, and political assessment. A more effective strategy should be proactive, focusing on prevention and early intervention. The challenge is that biosecurity systems are inherently complex, involving multiple actors, overlapping regulations, feedback loops, human behavior. But there are some clear steps that can be taken. First, quarantine protocols need to be strengthened. Risk mitigation can be applied at different points along transport pathways, especially in air passenger traffic. Screening during pre-departure, transit, and arrival phase is essential. Cargo pathways are also require stricter controls. More vigilance during packing and shipping would reduce stowaway incidents, but these measures are still poorly implemented in the UK. Another layer of complexity is climate change. Ecosystems must be prioritized based on the vulnerability, with high risk sites protected and those already under pressure actively managed to prevent further decline. In this context, regional approaches are more effective than broad global frameworks because they allow us to address specific pathways, species response, and knowledge gaps. And importantly, pathway management should take priority over species-specific control. By focusing on how alien species are introduced, whether through trade, travel, or cargo, we can reduce risk more effectively than trying to deal with species one by one. To conclude, the current number of wildlife stowaways may appear small, but with increasing globalization, they are likely to become a serious concern for UK biodiversity. Here also, the numbers might not be that big because, as I mentioned, there's a big knowledge gap and a lot of the stowaways are actually underreported. But this study shows that the problem is already growing and that early action is needed. In short, wildlife stowaways are not just a theoretical issue. They represent a very real and growing risk to biodiversity, ecosystems, and even public health in the UK. Acting early, managing pathways, and strengthening biosecurity are critical if you want to get ahead of the problem. Thank you.
So, hello everyone. Thank you for having me today. Thank you, Harpreet, for your amazing presentation. It ties in very well with my subject, so I'm very happy to have heard yours first. So, yeah, I've just finished my Master's by Research here at DICE with Jim Greenbridge, which you all know already. So my topic was patterns of beak and feather disease virus prevalence and effects on individual fitness in threatened island birds. So today I won't be covering the whole thing. I'll just go through some of the things I found in my second chapter, which is identifying strains of BFDV and their effect on recruitment of the Mauritius parakeet over 2 decades. So this chapter takes us to sunny Mauritius. It's an island in the Indian Ocean off the coast of Madagascar. So it's an island that has had a history of human disturbance, which has caused a lot of deforestation to the point where only 2% of the native forest remains. Obviously, that has caused an immense loss of endemic species, but there are some conservation success stories here too, some of which we've worked on here at DICE, namely the Mauritius kestrel, the pink pigeon, and the Mauritius parakeet, which is who we'll be talking about today. So this rather cheeky-looking Mauritius parakeet was once considered the world's rarest parrot in the 1980s, where it was believed that there were only 8 to 12 individuals remaining, of which only 3 known females. So this was a really dark time for the Mauritius parakeet. It was in trouble. So Gerald Durrell and Professor Carl Jones decided to step in to try and save the species from extinction. So in the 1990s, a decade of intensive management began to try and save the species. They put in nest boxes, feeding hoppers, they did predator control and even captive breeding to try and maximize productivity and save the species from extinction. And lucky for us, it worked. By the end of the 1990s, the population had reached 100 individuals. So that's it, right? That's it, happy days, perfect, all good. Well, like with all hands-on management, it's always a high-risk strategy, and there's usually some kind of complication along the way. And that complication happened in 2005, when birds started looking like this. So conservationist alarm bells started ringing, because obviously this bird has symptoms of cytosine beak and feather disease. So symptoms include feather loss, like this bird here, but also beak deformation, and ultimately immunosuppression. That is, if the bird is lucky enough to live that long, because this disease is very lethal for chicks, It's spread from mother to offspring, but also in the environment. So the birds will shed their feathers. The virus creates some kind of dust that goes everywhere in the environment, in the nest boxes, on the feeding hoppers, but also in the natural environment. So it's just really good at getting around. It's caused by beak and feather disease virus. So what is BFDV? BFDV is a single-stranded DNA circovirus. So it's very small. It only has 2 genes, the replicase and the capsid, and it's very fast evolving. So it acts more like an RNA virus, like COVID or Ebola. So, like I said, there was an outbreak in 2005-2006 where 41% of birds tested were found positive. So that called for the end of intensive management and the start of the monitoring period in Mauritius. Now, obviously, it's not to say that the intensive management wasn't worth it, otherwise the population would have gone extinct. So the conservationists did what they could, but as of 2005, they had quite, they put in some very strict biosecurity protocols to try and limit the spread of the virus because of us being there, basically, and still try to help the population recover. That is when they started sampling regularly, and that's why we've got an amazing data set of over 2 decades of sampling here. And actually, most of the samples are here at DICE, so we're very lucky to have those. And it's enabled us to basically look at the prevalence of the virus over time. So that's what you can see on the graph. And we've noticed that there are some subsequent waves since the 2005 outbreak, but the population has continued to grow. So that's the line on the graph. The total number of breeding pairs has continued to grow over time. So that's good. So that's when I come in. What was the aim of my study? So one of the questions that I was looking at was trying to understand how BFDV has affected the Mauritius parakeets population recovery. So one way of trying to do this basically was looking at the impact on recruitment into the breeding population. This is a metric that's really well studied by the Mauritian Wildlife Foundation, has been from the start. So we thought it was a good metric to, I'll just move that out of the way. Yes, so I thought it was a good metric to use, and we also wanted to look at levels of inbreeding. So obviously this population has lost a lot of individuals, so it's lost a lot of genetic diversity, which is quite important in this sort of situation. So we thought it was important to account for that in our models and include it and see if there was any interaction there. So that's how did I do it? So the lab work was undertaken by Pam before I arrived at the university. She extracted the DNA, did the PCR testing for BFDV in both genes, and that was sent to Macrogen. So Macrogen is in Amsterdam. Then they send us a very nice long sequence of the virus, and we collate that all together to form one data set per gene. And it looks something like this. It's quite hard to visualize the differences in viral strains when you look at something like that. So one nice visual way to do that is by creating haplotype networks. So that's what I did, one per gene. And basically, the larger the circle, the more the viral strain is present in your population. Once I'd created those, I added the studbook information on recruitment and inbreeding and everything else that I wanted to test and then didn't work. Those things worked. And we looked at, we'd created logistic regressions in R, looking at the effect of haplotypes on recruitment and inbreeding in the population. What did I find? So initially, when I created the graphs, I had a look, and there were some initial reactions basically from me when I looked at those. The first thing was the variation clearly between the rep at the top and the capsid at the bottom, and I wondered why the rep looked so conserved. I thought that it was just quite striking how different they were. And actually it's normal because the replicase is essential in viral survival. It needs to replicase to work well, otherwise it won't spread well to other individuals. So that's why it looks relatively similar in different individuals. Whereas in the capsid, there's more room for variation. So that's normal, which is good. And then I also saw there was a presence of single host haplotypes. So that's all of the small circles. Basically, those are only present in one bird. And I wondered straight away, why were they only present in one bird? Is it because the virus isn't working properly, so it's not spreading to other individuals? Or is it because it's really deadly, and so it's killing the bird off straight away and it's not spreading to other individuals? That's something that I personally wanted to look into when I looked at those results. And then the last thing is I noticed that the outbreak haplotype has almost no crossover with the most recent wave of 2020. So we can see that the virus has evolved quite a lot in the last 2 decades to look basically completely different to it when it was first appeared in the population. Right, yes, so in terms of the GLMs, we found that the impact on recruitment has become more severe over time, so it's had a negative impact on recruitment. And we've also found that the individuals with single host haplotypes, so the ones only present in one bird, were rarely recruited. So that seems to mean that those viral strains were very deadly, and that's why they're not being recruited into the population, basically. So that answered my question, so I was very happy about that. And then all of the birds that were less likely to be recruited were also more likely to be inbred. So we saw an interaction there, which we thought was interesting for building the story around the questions that we found, which is the graph that is just there. So that's great, but what does it all mean to someone that hasn't actually done this for a year? So as all good research, my research has led to more questions than answers. Now my next question is really, is the virus becoming more lethal, or is the host just becoming less resistant to the virus? It's likely a bit of both. As with everything, there's virus-host co-evolution. So in large populations, which work normally, it's sort of a race. The virus mutates and evolves to try and kill its host in a better, more quick way. And then the virus tries to adapt as quickly as possible so that there are minimal outcomes from the viral presence in the population. So that's what happens in a large population. But in a small population, small recovering population, like the Mauritius parakeet, like I said, obviously the bird has gone through a huge population decline, which has meant that there's a loss of genetic diversity. So sometimes the bird just can't adapt. It can't adapt quick enough, which is probably what happened in 2005. It couldn't adapt, and so the virus, there was a massive outbreak, and we all were very scared for the birds. And then we've seen sort of the long-term effect of that. So there have been subsequent waves, and every time, periodically, every subsequent wave, we can see that the bird hasn't managed to adapt, so it's peaked in the population. So basically, the host falls behind. I hope I'm not talking too quickly. So some take-homes from me. So if you weren't convinced that genetics were important for conservation already, I hope that you are now. Obviously, they're often not in conservation management plans, and we aim to change that with my supervisor. Yes, obviously, if we'd never done any sampling, any testing, we would have no idea what happened to the Mauritius parakeet, and we would have no idea how long the virus has had an impact on the population for. So we think it's crucial. We've been able, through this testing, to tell the story of the Mauritius parakeet in the last 2 decades, which is really rare, and it's a great example to take and help other people avoid these problems happening in other parrot species. It's also shown the long-term impact of emerging infectious diseases like BFDV. So obviously we've seen that it doesn't sort of appear and then go away. Once it's here to stay. And 2 decades later, we're still seeing the impact in the population, which is why it should be included in management plans, because it doesn't just go away. So you need to include it and work to try and avoid it happening, especially in a recovering population like the Mauritius parakeet or other populations, because BFDV is present in a lot of parrot species, but actually it's not always present in the management plans for these species, so that's kind of silly, because if we know that it might happen, then we can plan for it and try and mitigate problems that may come with that. So yeah, I think that's it. Thank you.
Evening, everyone. I'm Edith, as Jim has introduced me, and I'm going to share with you my research, which was entitled Understanding Community Perceptions on Lion, Panthera Leo Conflict Mitigation Strategies, and it was done in Nyami district in Zimbabwe from April up to May. And this is the outline that I'm going to follow. I'll first introduce and give a background of Nyami as well as my research project, then move on to the goal, aim, hypothesis, and research gaps. Then we'll move on to the methodology, study area, study design, approaches, data analysis. Then I will share my findings and recommendations, then I'll conclude. Lion populations have been decreasing over the past 40 years from approximately 100,000, according to research, to approximately between 20,000 and 40,000. Nyami district, located in Zimbabwe, is made up of farmers, mainly subsistence farmers, who survive on livestock rearing and crop production. And as a result, there is a lot of human and wildlife conflict taking place. And apart from that, there is also human-lion conflict happening. And most farmers are losing their livestock to lions. And as a retaliatory action, the farmers end up killing lions, and this has brought a decline in the population of lions in Zimbabwe. As a means to curb the reduction in populations of lions, different stakeholders have met and come up with different strategies to mitigate human-lion conflict. Amongst those, I was hosted by an organization called Wildlife Conservation, and to date, they've installed 224 lion lights, which are benefiting 56 households. And these lights provide safety, peace, and a sustainable solution to human-wildlife coexistence. And amongst the strategies, we have raised the crows that farmers are building to deter predators from attacking their livestock. We also have community guardians who embark on community patrols, and to date, 1,526 community patrols have been conducted, fostering coexistence and safeguarding lives and livelihoods. And as part of the role of the community guardians, they also educate the communities on how they can interact with lions. There is also lion collaring, where there is the white car there. The collars are being put on the lions to track their movements, and the community guardians advise the communities whenever the lions come into the, approach the communal lands. Then, as part of the strategies as well, there is use of the bomber sheeting or predator-proof mobile crows, which are being used by farmers to protect their livestock. And these mobile-proof bombers are mainly used during the night, since during the day, the cattle will be out foraging. According to my title, I said understanding perceptions of farmers. And in most cases, when people hear about perceptions, they think about beliefs. But perceptions are not only about beliefs. They are holistic. They look at observations, understanding, interpretation, and evaluation. And it's very important to understand what the communities want, and also to engage the communities so that we know what they understand about human-wildlife conflict mitigation or human-lion conflict mitigation. Because once we do not involve the communities, our conservation practices go in vain. The goal for my study was to identify the most effective preventative measure for replication in other lion populations across Zimbabwe. It is vital to note that it's not only Nyami facing these human-lion conflict challenges, but most communities living adjacent to protected areas are facing challenges with human-wildlife conflict, including the lions. And my aim was to understand farmers' perceptions of the effectiveness of various interventions that I've mentioned before, aimed at reducing lion conflicts, which is the Panthera leo. Then my objectives were to quantify social and ecological factors that influence lion attacks on human and livestock. The second objective was to compare the effectiveness of different mitigation measures in reducing lion predation on livestock. And the third was to evaluate whether and how different preventative measures and compensation schemes, if they are there, reduce lion attacks on both humans and livestock. And as part of the hypothesis, I assumed that raised crows and reinforced crows are more effective in reducing human-lion conflict as compared to lion lights and mobile bombers. Financial dependency factors positively influence the perceived levels of trust in conservation initiatives. And finally, social dependency factors such as community participation in NGO-led interventions enhance perceived relationship satisfaction of the local farmers. Then, as part of the research gaps, it's noted that there is limited use of the SET, which is the social exchange theory model, and Bayesian models in conservation studies. And today, as I stand here, I'm proud because I managed to use the Bayesian model and also the SET theory during my research, even though it wasn't easy at the beginning. Then few research papers by female conservationists on carnivore studies. Then moving on to the methodology, I'll start with introducing the study area, which is Nyami-Nyami. Nyami-Nyami is located in the northwestern part of Zimbabwe in southern Africa. The district covers approximately 456,650 hectares, and it consists of 12 administrative wards. And it has approximately 11,091 households with an estimated population of 45,774 people, according to the Zim Stats for 2023. Nyami-Nyami spans under agroecological region 4 and 5, which receives erratic rainfalls with high temperatures ranging between 20 degrees Celsius and 30 degrees Celsius, and sometimes it can even go beyond that. The area is more dominated by the Shona and the Tonga-speaking people. As part of the sources of livelihoods for the farmers, most people depend on crop production and small livestock rearing. And the district is a home to various species containing as well the big five, which is the elephant, lion, the leopard, the buffalo, and the rhinoceros. Thank you. The area borders two protected areas, which is the Matusha-Dona National Park. I think you can see it over here. Matusha-Dona National Park is under the KAZA, which is the Kavango-Zambezi Conservation Trans-Frontier Conservation Area, and as well as the Chirisa Safari Area. Chirisa is here. So you realize that there is a lot of movement of lions which occur from Matusadona National Park to Chirisa Safari Area, as well as the Chizarira National Park and Chete Safari Area. And this automatically means that as the lions move, here there are villages, and this is a heat map of the area where I did my survey, and there are people living there. Automatically there is a human-lion conflict because of that. Climate change has also induced the human-lion conflict within the area. And apart from that, lack of fencing materials to mark the boundary between wildlife area as well as the communal land has also contributed to a lot of human-lion conflict taking place. From the study area you will notice that there are many lion incidents from the star here, Watt 10, Watt 8, part of Watt 9, Watt 7, Watt 6, Watt 3, Watt 1. These areas are very much affected. And from my study design, I sought approval for ethics, then applied for a study permit, and also conducted courtesy visits to the gatekeepers. And as part of my study approaches, I used a mixed design approach where I conducted quantitative and qualitative methods. Then I used the structural exchange theory, which enables an individual to assess relationships between different factors. And this is the diagram of the set theory that I used. And these are the variables that I was measuring: communication quality, opportunistic behavior, social dependence, financial dependence, trust, relationship satisfaction, propensity to live, and commitment, in order to view how the communities are accepting the interventions within the area. For qualitative data collection, I used an open-ended questionnaire, and I managed to interview key stakeholders within Nyami Nyami. And for quantitative data collection, I used a questionnaire, used the Kobo Collect toolbox, 5-point Likert scale statement, and interviews. Then my participants were randomly selected from 38 villages and 9 wards. Then for the data analysis, I prepared my data, coded using ARA Studio. Then initially, I had planned to conduct a B11 analysis, but the model could not fit, so I changed to Lavan to analyze the data. So this is the whole process that I undertook in order to find whether the model was fit and good to the data that was collected. And these are the results, but I'll go straight to the findings and recommendations because of time. The first one, on the effectiveness of mitigation strategies, the mobile-proof boma came first from the mitigation strategies that are being implemented. Then the second one, communities felt that there is need for fencing materials to demarcate wildlife and communal areas. Then I also found out that the closer the communities are to the protected area, the more the conflict. Then instead of the lions, because initially when I chose my project, I assumed that lions are the ones which were causing more harm, but only to find out that it was the hyenas instead of the lions, and communities could not differentiate between the spores of the two. Then there is need for stakeholder engagement within the area, and communities were happy in terms of their involvement, because most of the community guardians are from the local area and are the ones who are teaching other people on human-lion conflict management. Then in terms of compensation, currently there is a bill which has been put in place by the government to see whether they can compensate when there are attacks that happen. And just to acknowledge, I would like to acknowledge the Bene Scholarship Scheme for funding my education here as well as the research. University of Kent, the DICE team, for the lectures and all the support. Nyami Nyami Rural District Council, they assisted me during the research. Then the government of the UK for sponsoring my research program, Beak Wildlife Trust, Wildlife Conservation Action, and my fellow colleagues. Thank you so much.